In the last video, we talked about the pupillary light reflex as part of the autonomic division of the peripheral nervous system. Let's move our discussion forward by talking about the other branch of the PNS, the somatic division, which governs voluntary movements. The somatic nervous system includes skeletal muscle, which are the muscles attached to bones by bundles of collagen called tendons, and excludes cardiac muscles and smooth muscle. So in the spirit of the NBA finals going on right now, let's look at basketball players as a prime example for voluntary movement, regardless of your opinions about LeBron and his highly publicized cramps. The real action, which makes all of this even possible, takes place at what's called the Neuromuscular Junction. Now the name kind of gives it away because we know that a junction is where two things come together. So a neuromuscular junction is where a neuron and a muscle cell come together at what's called a synapse. So I'll just quickly draw the basic anatomy of a neuron. So we have the soma, or the cell body. We've got these finger-like projections called dendrites, which receive signals. Then we have the axon, through which an action potential propagates. Then we've got the axon terminal, which is where the neuron ends. Now, the axon terminal comes really, really close to the muscle cell. So this is the muscle. And an action potential travels down the neuron and then is passed on to the muscle, causing contraction through a series of events that occur at the neuromuscular junction, or also known as the synapse or synaptic cleft. So let's take a closer look at the events that are occurring at the neuromuscular junction. So we have zoomed in here the neuron, then we've got the muscle cell. Remember, this gap is called the synapse, or the NMJ. And then on this top side, we have the presynaptic side, because this is where the signal is coming from. And then on this side is the postsynaptic side, or the receiving end. So within this neuron, we have these vesicles containing tiny little neurotransmitters called acetylcholine. We also have, outside of the cell, ions, calcium and sodium. And the extracellular concentration of these ions are much, much higher than the concentration inside of the cells, setting up this gradient. Now, ions want to flow down the gradient, so calcium and sodium really want to get into the cell where there's not that much ions present, but they can't because of the presence of these channels, which are closed when the cell is at rest. So on the neuron, in purple, we have voltage-gated calcium channels. And then similarly, on the muscle cells, we have what are called ligand-gated sodium channels. which are also closed when the cell is at rest. But what happens when an action potential comes along? It's propagated down and reaches the axon terminal. So when that happens, the inside of the cell becomes a little bit more positive, causing the voltage-gated calcium channels to open, allowing calcium to flood into the cell. When that happens, you get what's called exocytosis, when these synaptic vessels fuse with the outer membrane of the neuron and releases the neurotransmitter acetylcholine into the synaptic cleft. Now, this allows the acetylcholine to bind to special receptors on the sodium channels, causing the sodium channels to open, allowing sodium to flood down its gradient into the muscle cell. This causes an action potential leading to, ultimately, muscle contraction. So once muscle contraction is achieved, we want the converse to happen, relaxation. So how does this happen? Well, we have the axon terminal, we have the muscle cell, and I'll draw in some of these sodium channels with acetylcholine bound. So this is the contracted state, and if we want relaxation to happen, the acetylcholine will detach from these receptors, and then the action of this guy, acetylcholine esterase, will take over. Now in biology, when you see the suffix ase, think enzyme that cleaves. So lactase cleaves lactose, cellulase cleaves cellulose, acetylcholinesterase cleaves acetylcholine. Now, acetylcholine is cleaved, so this is the structure of acetylcholine. It's an ester, and when cleaved, becomes acetate and choline. Now, choline can be taken up back into the cell, combined with coenzyme acetyl-CoA to remake acetylcholine neurotransmitter, which can be released when the next action potential propagates down and reaches the axon terminal. So, what do you think happens if you block the activity of acetylcholinesterase with an inhibitor? So if that happens, you block the enzyme, you block the ability to remake acetylcholine, and what you end up with is that the acetylcholine neurotransmitter stays in the synaptic cleft longer, allowing it to continuously bind the receptors instead of getting broken down and recycled. This causes overstimulation of this neuropathway, leading to prolonged or repeated involuntary contraction of muscles, also known as tetany. So this is actually really dangerous and is what happens when you get a snake bite or come in contact with snake venom, is that it blocks the proper functioning of this enzyme and is actually really dangerous and can even lead to death. So that concludes our video series. I hope you learned a lot, and thanks for watching.